All right, at the top of this week, I wanted to spend a minute or two talking about molecular cloning. And so this is related to the material that you'll be working on in lab. So the idea is that we're taking the plasmids that we made before spring break, and we are transforming those plasmids, putting them into bacteria. So I wanted to talk a little bit about that process. So we have a vector here. That vector contains our origin of replication and then some kind of selectable marker. And so we talked in lab about how we're using the ampicillin resistance gene or the kanamycin resistance gene. And so one or the other would be present in the vector. So an origin of replication, that means this thing can be duplicated, and then a selectable marker means that we can eliminate bacteria that do not have the vector of interest. So we can select for bacteria that have the vector that we're after. So if we were cloning, we would take a DNA fragment. There would be sticky ends on either end of this. We would break this vector, so there'd be a sticky end there. We're breaking it using restriction endonucleases. The DNA fragment can then insert into the vector, and then we can use ligase to covalently bond that insert into the vector. And so I think y'all have heard that terminology from your lab instructors. Insert and vector was a big part of the DNA hybridization lab report. So this terminology should be pretty straightforward. What we have to do once we have this plasmid is we have to put it inside of a bacterium. So one single bacterial cell is called a bacterium. And so the idea is we're transforming this bacterium by placing the plasmid inside of the bacterium. And so we do that through a process that we're going to talk about as part of this video. But once the plasmid is inside the cell, the plasmid can copy itself, so it can replicate. We can end up with multiple copies of plasmid within the cell. And so often when we're using vectors, we use vectors that are called high copy number plasmids. And so the idea there is that high copy number plasmids are going to make lots and lots of copies. So each single bacterium will contain 15, 20 plasmids within that single cell wall. So that's the idea. We've got here a high copy number plasmid, so one bacterium, multiple copies of the plasmid. That cell can then divide and give rise to two cells. So division of that host cell creates two cells that both have plasmid. Eventually those can divide again. That happens multiple times, and then you end up with a colony. And so the colony is what results from one single bacterium. So any place one single bacterium can survive, it will divide and divide and divide and replicate and divide, and then you end up with this mound of cells where that one single bacterium was. So let's talk about this process. How do we get this plasmid inside of the bacterium? So this is kind of an old slide. You can see it's a little bit pixelated, but I think it does a pretty good job of describing this technique, and it's an old technique. So maybe it's appropriate that we're using some old looking graphics to explain it. So here we've got an LB agar plate. There are colonies on this LB agar plate. We can pick one of those colonies. We can grow it in. We can grow it up overnight in liquid culture, and so then we take that liquid culture and we collect those cells using a centrifuge. We dump off the media and we transfer those cells into cold calcium chloride. And so the idea here is that we're resuspending the cells in cold calcium chloride. The cold calcium chloride is going in and masking the charge that's normally associated with the membrane and the bacterium. It's also masking the charge that's normally associated with the plasmid. So neutralizing the charge means that those things that would normally repel each other can come into closer contact. And so here we're adding this ampicillin resistance gene positive plasmid to the mixture of cells. And then so we hope that what's happening in that scenario is that there are plasmids that are closely associated with those membranes. So then when we move the cells over to 42 degrees, we're heating them, we're raising the temperature from ice cold to 42 degrees, which is above body temperature. We're heating them up so much that what should be happening is the membrane should be moving, oscillating, bouncing around. The cell should be expanding as part of this as well. And so we're hoping that in that expansion, the cells, in the expansion and the movement of the membrane, the cells are taking up plasmid. So this is going to be low efficiency. We're not going to do it thousands of times. We're going to do it, I don't know, 10, 30, 100 times. So if we've got lots and lots of bacteria in here, only a few of them are going to be transformed. And then what we'll do is we'll take those bacteria, we'll put them on a plate, spread them out, allow them to grow for 24 hours, and the next day we should have bacteria that are transformed, meaning they're positive for the plasmid with that ampicillin resistance gene. And we can be reasonably sure that they're positive because they're growing in the presence of that ampicillin. So that's how the process works. You should be getting more information about that from your experience with the lab. The reason that we're doing this here is because I've got these slides put together and the lab videos were shot in front of a chalkboard. And so the media didn't quite mix right. I want to point out one more thing before we move on. And so up here, we're saying that we're taking cells that are in the log phase of growth. And so if this is the way the whole thing works, one of the requirements to get to the end point of having cells that have that ampicillin resistance gene is that we need to have cells at the beginning that are in the log phase of growth. And so on this next slide, I can show you what we mean by log phase. And so we could break up bacterial growth into 4 different phases. Here we've got the lag phase. This is the early stage of replication, or early stage of division. We're copying, but we don't have a ton of bacteria at that point. In the log phase, so this is exponential, right? Each bacteria is capable of giving rise to 2. Each of those bacteria is capable of giving rise to 2 more, et cetera, et cetera, et cetera. And so the log phase is where growth really starts to pick up steam. So here we've got cells that are growing exponentially, and they're at a point where they're very productive. So there's a high number of cells. Importantly, what hasn't happened yet is we haven't gotten to a place where the media is starting to be, the contents of the media are starting to be restricted. And so what happens eventually is the E. coli will use up all the resources that are available in the media, and the growth will start to slow. And so we'll get to what we call the stationary phase. And so the reason that we want cells in the log phase is because at that point, not only have we not limited growth, we also haven't induced death. So the cells are really healthy at that point. They're not being selected. There's not selective pressure on their survival. Also, they're not at very low numbers. So we've got high numbers of cells that are not yet being selected. And so this is kind of the ideal place to draw our cells for our transformation procedure. So that's what y'all need to know before you really get too deep in the lab. So yeah, reach out to me if you have questions. Thanks.